All right, here we go with part 3 of our digestive series of videos. So the stomach is a J-shaped enlargement of the GI tract. It connects the esophagus to the duodenum, which is part of our small intestine. Here we see the picture of a stomach. It has some certain parts to it that you should know about. Coming down off the esophagus, of course, was the lower esophageal sphincter. At the top here, this portion is called the fundus of the stomach. We have this whole outer portion is called the greater curvature. The inner portion of the curve is called the lesser curvature. Right after the esophageal sphincter, the bolus is going to move into the cardia, this section right here. Then there's the fundus at the top, and then there's this big portion inside, which is the body of the stomach. And then at the end, we have the pylorus. There's also the pyloric sphincter here, excuse me. And there's like different parts of the pyloric canal, the pyloric antrum, but really we just look at we don't really consider those too much. And then at the end, this pylorus sphincter leads towards the duodenum, which is the first part of the small intestine. You'll also see an interesting anomaly, I guess, for the stomach. It has 3 layers of muscle. We not only have the longitudinal and the circular, we also have the oblique layer. So here we see again, this is a cadaver one, the different parts of the stomach. Another thing I want to point out is that the stomach has gastric folds in the mucosa. So they're also known as rugae, R-U-G-A-E, rugae, but you can call them gastric folds. When you use an endoscope, if you've ever had an endoscopy, they'll look down your esophagus and possibly into the stomach, and you'll see all these gastric folds. They're important because they allow the stomach to expand in size. So when you eat the supersized meal from McDonald's, hopefully you guys don't eat it too much, but it'll expand that stomach to a greater size. We've all felt it, like when your stomach is like, oh, that Thanksgiving dinner, it's so full. So at that point, the rugae or the gastric folds are going to disappear because they've stretched out. So the functions of the stomach, we got 4 of them listed. It helps to mix the saliva, food, and the gastric juice with the bolus, and it forms a substance called chyme. So it's going to turn from a bolus into chyme. It serves as a reservoir for food before being released in the small intestine. So we eat much quicker than we can digest. So the stomach holds that food. It also secretes something called gastric juice. That contains hydrochloric acid, pepsin, intrinsic factor, and gastric lipase. So let's go through those. HCl, or hydrochloric acid, kills bacteria that we might have eaten. It denatures protein, so it changes the shape of protein, so that kind of starts the breaking down of proteins, and then activates the lingual lipase that was secreted in the mouth. Then begins the protein digestion. The intrinsic factor is necessary for vitamin B12 absorption. Okay, so you have to have this in order to get your B12 vitamin absorbed. And then gastric lipase is used in triglyceride digestion. So remember, fats are triglycerides, are the biggest form that we eat. And anything that is a lipase is going to break down fat. So lingual lipase, that was secreted in the mouth. Gastric lipase is secreted in the stomach. So gastric means stomach. Excuse me. And then lastly, the stomach secretes gastrin, which is a hormone. This is not an enzyme, it is a hormone. And that gets secreted into the blood, not into the cavity of the stomach, okay? So let's look at the histology of the stomach. It's pretty interesting. So we have the 4 layers, the mucosa, submucosa, muscular layer, and serosa. The epithelial layer extends down to the lamina propria, and it forms gastric glands and gastric pits. So let's look here. Here is the epithelial layer, and instead of being pretty much flat, like we've seen previously, it creates these long, it goes down all the way to the lamina propria. So it creates these are gastric pits here. And then also the gastric glands are way down here, okay? So the pits are just kind of like the starting of them, and the glands are down here. So there are several important cells in these gastric pits. We have all of these surface mucus cells, so you can pretty much know what that does. Secrete some mucus. We have mucus neck cells, which we'll get to these on the next slides. We have parietal cells. We have chief cells. We have the gastric glands down there, and then we have G cells. Okay, so we're going to get to those in just a minute. Excuse me. The submucosa, you can see that as it has the lymphatic and blood vessels down there. The muscular layer, like I mentioned, has 3 layers of smooth muscle. We have the oblique layer. You can see the direction in which the muscular fibers run are obliquely. The circular layer, which is going to be coming around the lumen, and then the longitudinal layer. We also have between these, the circular and the longitudinal layers, we have the enteric neurons for the muscular layer. So here's a close-up of the gastric glands. And remember, they're going to be responsible for secreting gastric juice. So gastric juice is the combination of the secretion from the mucus, parietal, and chief cells. What it is made up of is mucus, hydrochloric acid, intrinsic fat factor, pepsinogen, which we'll get to, and gastric lipase. Excuse me again. On the left here, you see a SEM of what the inside of the stomach looks like. It looks very cool. It has these surface mucus cells, but then you see how deep it goes into those gastric pits. Kind of neat. And again, we have the different cells, and let's go through them. Surface mucus cells, which are more or less near the surface, and also mucus neck cells, which are kind of on the neck. I think that's where it gets it from, the neck of this gastric pit. Both of them secrete mucus. We also have parietal cells. They secrete hydrochloric acid and the intrinsic factor. The chief cells secrete pepsinogen and gastric lipase. And the G cell is the one that secretes the hormone, gastrin. So if you could pause here and just watch sections 5.1 and 6.1, okay? And then come on back. So paracelsus is how mechanical digestion is occurring, excuse me, in the stomach. There are 2 parts to this. There is propulsion, which is the moving the gastric contents from the body of the stomach into the antrum due to peristaltic waves. So it's pushing it forward, or I guess it would be like through the stomach. The pyloric sphincter is usually closed. So when the mixture, this chyme, is pushed into the body of the stomach, or I mean into that pyloric sphincter, it gets pushed back into the body. So that's moving backwards, and that's called retropulsion. So propulsion and retropulsion mixes all the stomach contents back and forth, and then it results in this, the bolus turns into chyme, okay? And chyme is when it's a soupy liquid mixture. And then after we do this for a while, the particles get small enough, and then they flow through the pyloric sphincter, and this process is called gastric emptying. Now, the chemical digestion. The parietal cell secretion of HCl is stimulated in different ways. So how do we get that hydrochloric acid being secreted? It can come from acetylcholine, which is ACH, from the parasympathetic stimulation. So remember, we have the parasympathetic nervous system wanting us to rest and digest. So it actually causes parietal cells to secrete HCl. The G cells produce or secrete, not secrete, but release the hormone gastrin, and that causes the parietal cells to secrete HCl. And lastly, histamine from mast cells can also cause the parietal cell secretion of HCl. So having this acidic environment, so HCl, the hydrochloric acid, causes the environment inside the stomach to be acidic. What does that mean? Or what does it do? What's the purpose? First, it inactivates salivary amylase. So remember, we had that released in our saliva, and it was starting the digestion of carbs, of starch. So it gets inactivated. It also activates lingual lipase. So that was secreted in the mouth as well and traveled down, and now it's starting to work on breaking down the fats. The acid also kills microbes in the food. It partially denatures proteins, so starting that breakdown, and it promotes the flow of bile and pancreatic juice. So that's coming up. That's what's going to be from the pancreas and the liver and gallbladder. That is going to be put into the small intestines. So it's already preparing for the next step. It also converts pepsinogen, which we were releasing from the cells, into pepsin. So pepsinogen is not active until it hits the acid. And pepsin breaks down proteins. So it starts the digestion of proteins this way. Oops. Oh, my goodness, what happened? I don't know what happened there. Sorry about that. Went through the whole thing, didn't it? Okay. I apologize for that. So I want to make sure you understand when the chief cells secrete pepsinogen, it has to go into the stomach and hit the acidic environment in order for it to be activated into pepsin. This prevents the digestion of the chief cells because they're made up of proteins. So we don't want to eat them and break down our body. So instead, they can release that precursor, the pepsinogen, get it activated, and then start digesting the proteins that we ate, okay? And the stomach epithelium has alkaline mucus to protect it from that acid. So pepsin breaks down some of the peptide bonds between the amino acids, making them into smaller fragments. So remember, a protein has many amino acids together that are connected by peptide bonds. That strand then gets folded upon each other, and that makes the different categories of structure, I guess. So previously, we had some of that broken down a little bit because of the acidic environment. Then the pepsin comes along and breaks apart it into different amino acids. Now, gastric lipase takes triglyceride and breaks them into fatty acids and monoglycerides. The absorption in the stomach is very limited. The epithelial cells in the stomach are impermeable to most materials. The mucus cells can absorb some water, some ions, some short chain fatty acids, and certain drugs, including aspirin, as well as alcohol. Here we go through the different, you have some really good tables in your book that summarize a lot of this stuff. So I just wanted to include some of those. And they're in the PowerPoint. I just hide the cells for most of them. So we have what happens in the surface and neck, surface mucus cells and the mucus neck cells. They're secreting mucus. They also absorb a small amount of water and what we just mentioned. The parietal cells secrete intrinsic factor needed for the vitamin B12 absorption, which is used in red blood cell formation. Also, they secrete hydrochloric acid, which we went through a couple of times what the importance of that is. The chief cells secrete pepsinogen, which is activated to pepsin to break down proteins into peptides. It also secretes gastric lipase to split triglycerides into fatty acids and monoglycerides. The G cells secrete gastrin, the hormone, which stimulates parietal cells to secrete HCl, chief cells to secrete pepsinogen, It also contracts the lower esophageal sphincter, increases motility, and relaxes the pyloric sphincter. We have the muscular layers and the pyloric sphincter as well. Now, vomiting, also known as emesis, is the forcible expulsion of the contents of the upper digestive canal, so the stomach and sometimes the duodenum. Emesis is stimulated when there is stomach irritation or distension, unpleasant sights, general anesthesia, lots of things can make you want to throw up. This works when nerve impulses are sent to the vomiting center in the medulla oblongata. This then sends impulses back to the digestive organs, the diaphragm, and the abdominal muscles. The stomach is squeezed between the diaphragm and the abdominal muscles, and the contents are expunged. All right, another accessory gland is the pancreas, or accessory organ. It contributes to the digestion in the small intestine. It's found retroperitoneal, posterior to the stomach. It produces enzymes that digest carbs, proteins, fats, and nucleic acids. So basically all of our main groups of food. It produces sodium bicarbonate, which buffers stomach acid. And it empties its contents into the duodenum via 2 different ducts. So when this is the shape of the pancreas, they have different parts to it, like the tail, the body, the neck, okay? The head. We see the pancreatic duct is right here. We also see a bile duct, which we'll get to how that comes together with the pancreatic. So we're going to talk about the ducts between the pancreas, the liver, and the gallbladder, because they're all contributing their juices into the small intestine. So exocrine secretions leave the pancreas via 2 ducts, the pancreatic and the accessory duct. The pancreatic duct joins the bile duct from the liver and gallbladder to form the hepatopancreatic ampulla, which then is the entering point of the duodenum. So, excuse me, here's our pancreas. The pancreatic duct is shown here in brownish color. We also have the accessory duct. So the accessory duct just goes right in the duodenum, closer to the beginning of the duodenum. The pancreatic duct, which is the major one, combines with the common bile duct from the gallbladder and the liver. So we have this bile duct coming off of the gallbladder. See, the gallbladder has a cystic duct. The liver has a hepatic duct. They come together to create the bile duct, okay? The bile duct combines with the pancreatic duct, and we can see it over here on the right. Here's the bile duct, here's the pancreatic duct, here's the hepatopancreatic ampulla, which then goes into the duodenum, okay? Pancreatic juice. It's a mixture of water, some salts, sodium bicarbonate, and several enzymes. So the sodium bicarbonate causes the pancreatic juice to be slightly alkaline. So that's the complete opposite of the acidic stomach. Excuse me. That buffers the acidic nature of the gastric juice. It stops the action of pepsin, and it creates the proper pH for the enzymes in the duodenum. So digestive enzymes, we have to digest our starches or the carbohydrates. We have pancreatic amylase. For proteins, we have trypsin, chymotrypsin, carboxyl peptidase, and elastase. But these, again, are secreted in their inactive forms because we don't want the pancreas to get digested because it's made up of a lot of protein. So they're secreted as enterokinase. I'm sorry, they're secreted as trypsinogen, and the enterokinase found in the pancreas activates trypsinogen into trypsin in the small intestines. I'm sorry, the enterokinase is secreted in the small intestine. So in the small intestine, we have trypsinogen being activated in trypsin. Then the trypsin activates the other 3. We also have to digest the fats or the triglycerides. We have pancreatic lipase. This is our primary lipid digestive enzyme. So this is much more important than the lingual lipase. And then to digest nucleic acids, we have ribonuclease and deoxyribonuclease. The liver and the gallbladder are two more accessory organs. The liver is our second largest organ in our body after our skin, and it is the heaviest gland that we have. So we see the liver here. It's found in the right hypochondriac region of the abdominal cavity. There are two lobes. The right lobe is larger than the left lobe, and it's separated by the falciform ligament. So that, again, is that fold in the mesentery. The gallbladder is this little guy here. It's a pear-shaped sac on the posterior surface of the liver. The liver makes bile, which is important in the emulsification of fats. And the gallbladder stores bile until it's needed. And again, here we have the different ducts and how that bile is makes its way into the duodenum. So in the liver, we have the right and left hepatic duct and the common hepatic duct. It forms a common hepatic duct. The cystic duct comes to that, and we end up with a bile duct. The bile duct then comes in with the pancreatic duct, doing that hepatopancreatic ampulla, and then into the duodenum. So the liver has a really interesting histology, and you guys will be covering that in lab as well. So the cells that make up 80% of the liver are hepatocytes. They form three-dimensional hepatic lamina, and they secrete bile into the bile caniculi. So if we look at this picture right here, it has all of these little cells are hepatocytes. The hepatic lamina are what is created when you get a bunch of these together. And then in between them, we have this, we'll look at the closer one up here to see what we have. But there's a central vein here, and coming off of the central vein, we have these hepatic sinusoids, okay? If we look at a close-up here, excuse me, these are the hepatocytes forming the hepatic lamina. In between them are these small ducts that are bile caniculi. There's small ducts between the hepatocytes that collect the bile that's secreted by the hepatocytes. And then the bile travels to the bile ductules, which are bigger, and then the bile ducts. So let's look at this picture here. We have the hepatic caniculi. You can see them right there. And then they're going to go through the hepatic, the bile ductules into the bile ducts until you get some big bile duct, and it can be traveling that way. Now, hepatic sinusoids are highly permeable blood capillaries between the hepatocytes, and they have oxygenated blood from the branches of the hepatic artery and deoxygenated blood from the digestive organs and the spleen. And that comes from the hepatic portal vein. So we talked about the portal circulation previously. So if we look at the blood vessels, excuse me, it's shown here in blue, okay? So we have a mixture of oxygenated and deoxygenated blood. See how we have the hepatic artery and the hepatic portal veins. We have oxygenated and deoxygenated blood. Now the blood flows in the opposite direction that the bile flows, okay? So when it comes in through here, it delivers the blood and it goes to the central vein here and then to the hepatic vein, okay? Now in here, we have these stellate reticuloendothelial cells. They're also known as hepatic macrophages or Kupfner cells. So those are seen here, these like yellowish cells that the blood is going to travel through. As the blood goes through this, they destroy the old white blood cells and red blood cells and any bacteria that's found in the blood. So this is kind of the filtering action or the cleaning of the blood that occurs. It's done by these cells, okay? So a lot going on in the liver. Again, this is how the blood comes into the liver. We have oxygenated blood from the hepatic artery, and then we have nutrient-rich deoxygenated blood from the hepatic portal vein. It gets mixed in these hepatic sinusoids, and it's going to be cleaned by those Kupner cells. It then flows to the central vein, the hepatic vein, and to eventually the inferior vena cava, and then the right atrium of the heart. Now, the liver also has these structures called the portal triads, okay? And you can see it best down in this picture here. The portal triad is made up of three things, so like tri meaning three. We have a bile duct, a branch of the hepatic artery, and a branch of the hepatic vein. So here we have the bile duct. This is the hepatic portal vein and the hepatic portal artery. Okay, that's a portal triad. All right, gallbladders. Gallbladders also have rugae, or these mucosal folds. That allows for them to expand when they store more bile. Smooth muscle contractions ejects the bile into the cystic duct when it's needed. And it also not only stores the bile, but it concentrates it up to 10 times. There are problems called gallstones. This forms from the insufficient bile salts or lecithin or excessive cholesterol, and it crystallizes into stones. They can be from the size of a grain of sand to golf ball size. Treatment includes stone dissolving drugs, lithotripsy, or the removal of the gallbladder. Bile is a yellowish, brownish, or olive green liquid with a pH that is slightly alkaline, 7.6 to 8.6. Mostly water. It also has bile salts, which emulsify fats, cholesterol, lecithin, which is a phospholipid, bile pigments, mainly bilirubin from the breakdown of red blood cells, and ions. Bilirubin will break down in the intestines into stercobilin, which gives species the brown colors. Bile salts. What bile salts do is they take a large fat globule. Bile salts come and they break it into many small globules. And when it does that, it increases the surface area. This allows the pancreatic lipase to digest the fats more quickly because now there's lots more surface area. And bile also allows for the absorption of lipids once they're digested. Other functions of the liver and gallbladder. So for carbohydrate metabolism, they help to maintain the normal blood glucose level by storing glucose as glycogen or converting glycogen back to glucose. This occurs in the liver, okay? Excuse me. So the liver can allow for either storing more of that glucose as glycogen, or when we have low blood glucose, converting it back to glucose and putting it out in the blood. For lipid metabolism, the hepatocytes can store some triglycerides. They also can break down fatty acids, synthesize lipoproteins and cholesterol, and make bile salts by using that cholesterol. Proteins, for protein metabolism, they can deaminate the amino acids for ATP production. And they also synthesize all the blood plasma proteins. The liver also detoxifies drugs, alcohol, and hormones. It secretes or excretes bilirubin, synthesizes bile salts, stores vitamins and glycogen, phagocytoses, old red blood cells, white blood cells, and bacteria, and it activates vitamin D. And this is the end of our video for part 2.